Thank you. When you applaud like that, you almost make me feel like an artist. Thank you so much. So, welcome again to Science Meets Music. This is our signature public event that emphasizes the close link between science and the arts. How both of these endeavors arise from creativity, dedication, and passion that drives exceptional accomplishment. For those of you who don't know me, and I think there are probably very few in the room at this point who don't know me, I'm David Fitzpatrick, CEO, Scientific Director of the Max Planck Florida Institute for Neuroscience, located right here in Jupiter. And I don't know about you, but it's been a tremendous season. It's gone by in a flash. And I hope you have enjoyed it as much as we have. Tonight's Science Meets Music is the last of 2019. And it promises to be a very special night for all of us. Part of what makes it special is that we have a Nobel laureate with us tonight. Edvard Moser, who received the Nobel Prize in Physiology or Medicine in 2014, is visiting us from his home in Trondheim, Norway. Actually, he just told me he flew in from Shanghai. So this is actually Edvard's second visit to Science Meets Music. See, we have a program that attracts them all the way from Norway, all right? And I think he might have been attracted to the beautiful woman who's actually sitting next to him in the audience tonight. So if you've been to our previous Science Meets Music events, you know that I like to focus on a theme for my welcoming remarks, one that mirrors elements that you in the audience will experience during the presentation and the musical performance. And tonight, the theme is family. Now, in this modern world, family can mean many things. Those we hold dear to us are not necessarily located right down the street anymore. And this is the case for my family. And that is why I am so pleased to be able to welcome my son, John, his lovely wife, Soo Yin, and my 2-year-old grandson, Aiden, who are visiting all the way from Shanghai, China. They are with us tonight. I just heard that Aiden went to the Science Museum in West Palm today. And have you seen the brain exhibit there? You should go check it out. It's a wonderful exhibit, and my 2.5-year-old grandson, if you ask him what's in the brain, he says neurons, okay? Get him going young, okay? So, now, you might be wondering, why are they visiting? Why would they come all this way? Well, they're here to welcome a wonderful new member of the family, Deborah Felber. I am delighted to announce that Deborah and I exchanged our wedding vows this past weekend. Lots of things happening, okay? Now, family is tightly interwoven in both the lecture and the music part of our evening as well. Our speaker tonight is Hyungbae Kwon, one of our very own research group leaders at Max Planck Florida. Hyungbae joined the Max Planck Florida just after we opened our facility in 2012. And I'll say a bit more about this later, but suffice it to say, Hyungbae was a great catch for the Institute and for the Max Planck Society. He built a research program that has led to fundamental new insights into brain circuits and brought him prestigious awards and grants. But like many of us have experienced with our own family members, Hyungbae will be starting a new chapter of his life in another city next month, when his lab moves to Johns Hopkins University in Baltimore. Yes, we will miss him and everyone in his lab, But the feeling of loss is balanced by an incredible pride and support for the advancements he will make for neuroscience going forward. Now, another thing that makes tonight especially memorable is that both the science and the music are being brought to us by the Kwon family, okay? Hyungbae and his wife, Sanji Kim, have raised 3 wonderful daughters, and Hyungbae Kwon is an accomplished musician, having played the violin from the age of 6. Now, the move of the Kwon family to Florida made it possible for her to attend the Dreyfus School of the Arts, a fantastic place for young minds to pursue their passion for the arts and for the sciences, a point that Alex Dreyfus has emphasized for all of the years that I've known him. It's not just the music, it's the music and the science. Alex Dreyfus is with us this evening, as he is for many of our events. And I'd just like to take a moment to recognize his exceptional contributions to the cultural and intellectual development of our community. Alex, thank you. Now, Seo Young's musical talents led her to become concertmaster of the Dreyfus School of the Arts Philharmonic and String Orchestra, and she's received high marks and many accolades from the Florida Music Education Association as a member of the All-State Orchestra. But Seo Young also has a strong interest in science. She's participated in the Institute's summer high school internship program, learning how to use Nobel laureate Stefan Hell. You all remember Stefan, right? You saw his presentation here. She learned how to use Stefan Hell's super resolution microscope, and this interest led her to acceptance at Johns Hopkins, where she'll be a freshman in biomedical engineering this fall. Another example, yes, that deserves applause. Another example of how science and music can be brought together to achieve a wonderfully fulfilling life. Now finally, we'd like to take a moment to honor Bob Goldberg, head of the Benjamin School. Sadly, Bob passed away from cancer recently. From the time Max Planck Florida came to Jupiter, Bob was a friend to the Institute, and he welcomed us to this beautiful auditorium when our lecture series grew and needed a new home. Because of Bob, not only were we able to forge a relationship around science meets music, but his leadership and personal commitment to his students elevated the Benjamin School to new heights. And this includes a signature science program and a brand new STEM building on the campus. Tonight's performance and lecture is dedicated to Bob Goldberg, and we thank him for the service and the love he showed to our community. Once again, thank you all for being here and for your support of the Science Meets Music and the Max Planck Florida Institute for Neuroscience. Now join me in welcoming to the stage Hyungbae Kwon on violin, who will be accompanied tonight by another exceptionally talented pianist, Karina Inoue, a native from Brazil, graduate of the New England Conservatory, and recipient of a number of awards for her passion as a collaborative pianist. a talented family, indeed. So it is now my pleasure to introduce tonight's speaker, Hyungbae Kwon. Hyungbae is from Korea. He grew up there, attended college there, came to the US in 2002, to pursue his PhD in neuroscience at Albert Einstein College of Medicine, and then went on to his postdoctoral training at Harvard University. And as I mentioned, in 2012, we were very fortunate to recruit him as a research group leader at the Max Planck Florida Institute for Neuroscience. He was remarkably effective in setting up his laboratory, I must say, recruiting great students, excellent postdocs, and continuing to explore his passion in activity-dependent maturation of cortical circuits. And he initiated a novel line of research that you'll be hearing about tonight that has generated exciting new tools for the study of neural circuits that have had a huge impact on the field. Now, beyond being an exceptional scientist, Hyungbae, I know this term is used a lot, but he's really a nice guy, okay? He's quiet, he's unassuming, no drama. Some may even say a little shy, but I can tell you there is nothing shy about the way this guy approaches his science, all right? I've said this before, it's a little bit like Clark Kent and Superman. You know, Clark Kent in terms of his daily interactions, but boy, get him in the lab, and this guy takes off. He flies to new heights, delivering fundamental new insights into brain circuits. While he's been at the Institute, he's published 8 papers in high-impact journals. He received a number of awards, and one that we are most proud of is that Hyungbae is the recipient of the 2018 NIH Director's Pioneer Award, one of 10 scientists in the country to achieve this distinction last year. And this is an award that covers all of the biomedical sciences, okay? By the way, 3 of these awards have been made to scientists in Florida. Do you know where those scientists are working? 2 of them at Max Planck, Florida. The other is Ryohei Yasuda, I think, who is here tonight. And the other is from Scripps, Disney. You know, I think that says a lot, that our campus is where this level of scientific excellence is housed. Now, thank you for that. Tonight is actually a bittersweet moment for us. Because the Institute is actually saying goodbye to Hyungbae. In fact, after the conclusion of this program, the group leaders and the directors will be going out to celebrate Hyungbae's departure. Now, that might sound a little strange, right? So I think I need to clarify exactly why we're celebrating his leaving us. You have to understand what a group leader is. A group leader is they're exceptional young scientists who are recruited into the Max Planck Society and given an opportunity to launch their research program under ideal conditions, provided with guaranteed annual funding, core facilities that are not available in many institutions, guidance from internationally renowned scientists, an environment that attracts excellent students and postdocs, and as you can see, produces excellent science and attracts significant grant support. It's really, I would argue, a fantastic training ground for young scientists. And there are only 2 things that are expected. 1, they're expected to do exceptional science. And 2, after 5 to 9 years, they move on to a position elsewhere in a university or some other scientific institution. That's right, Max Planck, the Max Planck way is to create an environment that builds exceptional young scientists and then when they're successful, sends them away. Okay, I'm sure you know, there are a bunch of business professionals in the audience that are shaking their heads about the wisdom of all of this. You invest in them, they're successful, and then you send them away. But that is the Max Planck way. That's the way it happens in Germany, and we do exactly the same thing here. You should recognize it's a wonderful way to broadcast the research fundamentals that have made the Max Planck Society so successful over the years to the broader scientific community, contributing to the growth of scientific excellence and the next generation of scientific progress. In fact, Hyungbae joins two other research group leaders, Sam Young, you may remember Sam, and James Schumers, both of whom have moved on to new faculty positions. So we are celebrating Hyungbae's success and celebrating the fact that Hyungbae is leaving to become a Blue Jay. Blue Jay is the mascot of Hopkins. All right, I have to admit I didn't know that either. And his new title will be Assistant Professor in the Solomon H. Snyder Department of Neuroscience at Johns Hopkins. Please join me in welcoming another success story of the Max Planck Florida Institute.
All right, can you hear me? Okay, oh, good. Okay, first, I'd like to thank David for the amazing introduction. And before I start, I have to mention that I wouldn't achieve anything without having this generous support from David and Max Planck Florida Institute. I was really lucky to be working in this Max Planck Florida Institute, and I will really miss this amazing research environment of here after I moved to Johns Hopkins. I also want to mention that it's a really great honor for me to share my research here today to all of you. Thank you all for coming. And it's also a great opportunity to stand in the same podium with my daughter today. It will be a really great memory, which I will talk about today. So today I'm going to talk about what I've been doing at the Max Planck Florida Institute for the past several years. And mostly I'm going to talk about the new technology that we recently developed a few years ago, and we are continuing those research. And I hope you feel some excitement about the neuroscience research at the end of this talk. So I've been studying learning and memory for many years. About 15 years now, and basically learning is brain changes. For example, when you learn something new, that means that there is a corresponding brain circuit changes that is long-lasting, so you are able to use this new information in the future and again and again. So if you want to understand the learning and memory, we have to look inside the brain and what is changing during learning. Basically, that's what I'm doing, and there are many, many ways to study this learning and memory. Luckily, technology was developed, and this technology allows us to visualize the individual neurons in a brain. You see, we can visualize a well-isolated neuron here, and you see a neuron has a big cell body, and then it branches. There are many, many branches to communicate with many other neurons. If you zoom in a little bit more, we can see even detailed structure. You see these many branches are coming out from the cell body. And if you look even closer, you see very small, tiny circular structure here. And there are many of them along these branches. This small structure is called dendritic spine. And this dendritic spine is a structural correlate of a single synapse. What it means is, in a brain, there are many neurons and many neurons. And to make our brain functional, those neurons have to talk to each other. They have to communicate each other. And this dendritic spine is the place where this communication is occurring. In other words, another neuron makes a synaptic contact to this neuron on this dendritic spine. And typically, this pyramidal neuron in cortex has about 1,000 dendritic spines. That means this neuron is connected with another 1,000 neurons. Okay, so during learning, when we learn something, those neuronal connections are changing. So by looking this structure and function of individual dendritic spine is so critical for understanding mechanisms of learning and memory. And what people found in a previous study is this. So when animals learn something, people image the individual dendritic spines in a brain, And you see, people found that the new dendritic spine is formed. You see, before learning, there was no dendritic spine here, but after learning, these two dendritic spines were formed. So based on this observation, we can think that learning is a new connection because this new dendritic spine indicates a new connection. So when we learn something, that means our neuron is making a new connection with another neurons to get new information. And I was one of people who is interested in this dendritic spine structure and function. And when I was a postdoctoral fellow at Harvard Medical School, I asked myself what is the most important question about this dendritic spine. And I thought that the most important question is to understand how this dendritic spine is created. If I understand what makes this dendritic spine, that means I may be able to understand the core principles underlying these neuronal connections, and ultimately we will understand and learning and memory. So to do that, luckily, I had access to the cutting-edge laser technology. So we can deliver the laser into the brain in a very small area. You see, this is 1 micrometer, even smaller than 1 micrometer area. We can deliver the small laser beam into the brain area. Then together with specialized chemical, we can deliver the neurotransmitter in very local area in an arbitrary fashion. And when we deliver neurotransmitter in very local area, I was able to successfully generate new dendritic spines. In addition, I, for the first time, reported that first time shows that the entire process of new dendritic spine formation from beginning to the end. So when we deliver the neurotransmitter in local area, there was no spine, but the spine is growing, and I captured those events in real time in the brain. And this research actually brought a lot of big excitement in the neuroscience field, but I felt at the same time, it's not enough, because just looking tiny structure of a neuron may not be sufficient to understand the big picture of learning and memory. So after I moved to Max Planck Florida Institute, I initiated a new type of research. So I start to image behaviors of many, many neurons. For example, in this experiment, we are training this mouse on this floating ball, and then we are imaging hundreds of neurons activity in real time when mice are performing this task. For this task, we divided this ball into 4 quadrants, and individual quadrant, we put different texture on the surface, and mouse can sense this different texture on the surface of ball, and mouse try to find the hidden spot where we provide a water reward. So I'm going to show the example of this video. You see, this mouse is controlling this ball to find the hidden spot to drink water. You see this mouse run on the floating ball, and then whenever mouse hit this the hidden spot area, we provide the water, and mouse drink water. So we trained this behavior for over several days, and mouse learn this task easily, and learning this type of task. At the same time, we want to understand how this type of learning is represented in the brain. So in real time, when mice are performing this task, we are imaging the neuronal activity in the brain from the hundreds of neurons. So current technology allows us to visualize all the individual neurons' activity in real time when mice are performing this task. You see neuronal activity come and go in a very dynamic manner and looks kind of quite random fashion, but we are imaging this same population of neuronal activity over and over for several days, and we try to understand how this activity pattern is shifting when mice are acquiring this type of learning. And this is really powerful and cutting-edge technology. Not many people can do this type of research as today. But at the same time, I still feel these are not enough because of this reason. Brain has too many neurons. Even if we open the small window, even less than 1 millimeter window, there are already hundreds of neurons inside of that tiny area. and entire brain has too many neurons, and I'm just showing the example of a mouse brain. To help your understanding, I'm just comparing the human brain and mouse brain. And human brain is much bigger than mouse brain, and human brain has 10 billions of neurons. So by looking the behavior of hundreds of neurons, can we really understand the entire neuronal population? That's nearly impossible. For example, we about 100 or 200 people here, and by just examining the behavior of people in this auditorium, we will never understand the entire human population behavior of the world. So how we solve this problem? And of course, this is a very difficult question, and we may not achieve this type of task very soon. But at least we can think in something in a different way. For example, I'm showing one example. The first flight was actually recorded at 1903 by Wright brothers, and you know everyone, these Wright brothers first made this airplane fly, and it's official record. And at that time, 1903 is more than 100 years ago. At that time, they don't have a clear understanding about the physics and engineering background of the airplane. Without knowing all this fundamental physics and engineering background, they still make airplane fly. So in a similar way, we can make something work first, and then we can understand it later. Actually, by making this airplane, this trial and error based this development accelerate the understanding of airplane. So what if we control the learning and memory first, and then understand it later? So that was my idea, and I got some hint from this taking pictures. So when you go a nice place, or when you meet your good friend, or when your family get together, today's theme is the family, you take pictures, right, to have a memory. And once you take pictures, and several years later even, when you look at these pictures again, then your memory record and retrieved, and you remember what was happening at that time. So exactly the same way, I try to capture the memory neurons, and then a few days later, I retrieve this memory just by shining the light. So the way it works is we named this technique NK-Light, and K-Light is basically a technique that captures and controls active neurons by delivering light. I'm going to show a couple of examples so you will understand easily later. So basically, I'll give you an example. For example, when you throw a ball, then this throwing a ball relevant neurons will be active in our brain. And then we shine light to label this ball throwing relevant neurons. And once we labeled, without touching any other neurons, only we labeled this behavior relevant neurons, and later on, we shine light again, then we actually engineer the specialized protein to be expressed only in the labeled neuron. So later on, we can control this activity of a labeled neuron. We can suppress those labeled neuron activity, or we can increase the activity of those labeled neurons. In that way, we can control animal sensation, perception, or behaviors, or even emotions later. So I'm going to give you a couple of examples. So as a first try, we wanted to label this level press and water reward associative learning. So we trained this mouse to press level to get the water reward. And first step is labeling. So we shine the blue light. Whenever mouse press level and get water reward, we shine the blue light to capture those level press relevant neurons. And later on, we shine yellow light to selectively suppress those labeled neurons activity and see whether we can erase this memory or not. So that's how it works, and you will immediately understand by just looking this video. You see here is a mouse in this big chamber, and here, the lever is here, and the water come out at the center. And once we train this mouse learn this task very well. And mouse press this lever a lot to get the water reward. And we shine the blue light. Now this lever press and water reward relevant neurons are active, and we want to label those neurons. And once the mouse learn this task, now we shine yellow light, see whether we can erase this memory. So this mouse has been going through all the training, and now we shine the yellow light to see whether we can suppress this behavior. And mouse still press the lever, and water come out, but it seems this mouse forgot what this lever was for. So we successfully shows that we can control this animal behavior by using this type of technique. And not only we can selectively inhibit or suppress this type of behavior, we can even trigger the behavior. We can make animal move as we want. So the way it is, we also developed a kind of a very similar technique, but we named this technique iTango2. And this technique allows us to selectively label dopamine activated neuron. And its previous research already reported that whenever animal initiate the movement, dopamine is released, and dopamine make this movement initiation. So we selectively labeled those running and movement relevant neurons in the brain, and we directly activate those neurons, and we see whether we can even trigger this running behavior. So we put this mouse on this floating ball again, and we directly activated the running relevant neurons. You see, when we shine the blue light, and immediately after we shine the blue light, mouse start to run. And when we turn off this blue light, then mouse stop running. And if you shine blue light again, then mouse will start to run. And this is a kind of a good way to make people exercise, I think. So you will see mouse just sit and take a break, but we shine the light again. And immediately after, mouse start to run. And when we turn off light, and mouse stop running. So we also show the strong evidence that we can control the behaviors in a suppressive manner or activating manner. So now I'm more curious. So I want whether I can control even high cognitive level of brain function. So for this experiment, we designed this type of behavior. So we put this mouse in big circular platform in the big box, and then we provide several visual cues. And on top of the box, we put a big computer screen, and then we display this growing dark circle looming signal. And then mouse feels scared, and mouse escapes the shelter. So I'm going to show one example. So here is mouse, and here is the shelter. And we display this looming signal, or we also play a sound, then mouse feels scared and escape to the shelter. And this escape behavior is memory-based. So if we, to know this escape behavior is memory-based, in this experiment, we removed this shelter. So there was a shelter here, but we removed this shelter. And then when we delivered the threat signal or sound, then mouse feels scared and immediately escape to the previous shelter location because mouse remember, oh, there was a shelter. So I'm going to show one example. You see this blue circle indicate the previous shelter location, and now there is no shelter, but when we play the sound, mouse remember the location and mouse immediately escape to the shelter. So this is clearly memory-based, and to convince you this is really memory-based, so I'm going to show this experiment. So in this experiment, we performed a little more difficult experiment. So instead of removing shelter, now we relocate the shelter from here to here. In this case, mouse has a two choice. Mouse can escape to the previous shelter location by memory, or mouse can just escape to the new shelter location by just looking, oh, shelter there, I escaped there. You see, and see what happens. So this is the now current shelter location, and we relocated this shelter now here, and now this is a previous shelter location. You see, mouse immediately escaped to the previous shelter location, although there is a shelter here. So maybe I can play one more time. Deliver threat signal, mouse don't know where to go, and immediately escape to the shelter. So based on this experiment, we clearly know this is the memory-based. And we labeled this shelter-related neuron and see whether we can erase this type of memory. So again, we use the cat-like technique, and we selectively labeled when mice are sitting in the shelter. Once we labeled, and then we shine the light to see whether we can erase this type of memory. And first, to make sure, without shining light, just the threat signal, and see whether mouse has a normal escape behavior. So we are delivering threat signal. You see, mouse escape behavior is normal. Now we are shining the yellow light to selectively erase this memory. We are shining light, and we are playing this threat signal. And even if we deliver this threat signal, mouse is not threatened or even don't know where to escape. Even if mouse sees the shelter, it didn't escape. So successfully, it seems like we erased this type of memory, but that's not our final goal. Our final goal is to selectively label those shelter neurons, and then by directly activating those neurons, whether we can control mouse move this way or this way, even without any threat signal. So this will be possible or not. I'm going to show you an example, and I think you will like it. So now here there is no even shelter and no threat signal, and we are directly activating those shelter-related neurons. Wherever mouse stay, whenever we shine the light, mouse immediately escapes to the shelter. I show the one more time. So shine light and escape the shelter. I'm not delivering any sound or looming signal, just directly activating shelter neurons. We can control mouse movement direction, just activating those neurons. Isn't it cool? Yeah. so science is very fun, yeah. So I'm going to finish my talk by showing this slide. We are currently living in an era of mental disorders. And we are suffering, many people are suffering mental disorders like psychiatric disorders such as schizophrenia, bipolar disorder, depression, and OCD. And this number of the types of mental disorders are rapidly increasing these days. On top of that, we are actually surrounded by all kinds of mental problems such as like worrisome, jealousy, dejection, despair, anger, sadness. Those are the really ones that are bugging our life all the time. And without those kind of emotional feelings, our life will be much, much happier. And believe it or not, almost nothing is known about this type of brain actions. And we don't even know where to start, where we have to look at, relate to this type of behavior, of feelings. Now, using this new tool set, we can at least begin to look at what's happening this type of brain actions. So listening good music and doing exercise, meditation may not be only solution. So in the future, you never know, you just shine some light a few seconds. Thank you for your attention.
to say after that. That was absolutely wonderful. Another round of applause for the musicians. And a special thanks to the Kwon family for the entire Science Meets Music this evening. That was wonderful. Hyungbae, would you come up here, please, for a second? We have a ritual. They also get it. Thank you for your performance. And thanks to all of you for joining us for the Science Meets Music this year. And we hope that you have a wonderful rest of your year. And we look forward to seeing you back here next year for a number of exciting lectures about the brain and some absolutely beautiful music. Now, I have to make one more announcement, and that is, if you happen to go to the bathroom here and you notice you're missing your keys, please see Miriam. She is the flower lady that you saw up here. She should be in the back somewhere. She has found some keys that you might need to go home. And safe travels.